Okay, so now what I'd like to do is talk a bit more about the neuroscience of the disorder, but to move from talking about the neurochemistry of the disorder, so key neurotransmitters such as dopamine that are likely involved in the disorder, and to begin talking about the actual structure of the brain and how that changes in people who have schizophrenia. In order to understand the structure of the brain, you of course need to know the major landmarks, and we've already talked about several of these landmarks. So again, I would just like to remind people that we've got four major lobes that make up what are called the neocortex or the new cortex, which is on the surface of the human brain. And they again consist of the frontal, parietal, temporal, and occipital lobes. These lobes actually surround a large space which are known as the ventricles of the brain. There are four ventricles of the brain, the lateral ventricles, which are also labeled ventricles 1 and 2, and then ventricles 3 and 4. Ventricle 3 is located around the level of the thalamus, and ventricle 4 is located just below the cerebellum. These ventricles are important because they represent a space in the center of our heads, and we have fluid that's transmitted through this space called cerebrospinal fluid. It is a fluid that bathes the brain, provides the brain protection, carries important waste products out of the brain, and brings nutrients to the brain. So you can think of the brain as consisting of this overlying neocortex, consisting of four lobes, a large series of spaces known as the ventricles, and then a variety of subcortical structures that we've talked about: the limbic system, amygdala and hippocampus, the basal ganglia involved in movement, the midbrain, and the brainstem and the cerebellum. So as we talk about the structure of the brain, we'll be talking a lot about these major sort of large neural structures within the human brain. Neuroimaging in many ways has revolutionized the study of schizophrenia. While recent neuroimaging techniques, such as magnetic resonance imaging or MRI, emerged more recently in the 1980s, but if one looks at the history of neuroimaging, studies of neuroimaging in fact go back in the medical literature to the 1930s. With the development of the X-ray, one of the first types of imaging was a type of imaging in which the brain was X-rayed, but it was X-rayed after a bubble of air was inserted into the central nervous system. These studies were known as air studies, and they were first conducted by the neurosurgeon Walter Dandy. What Dandy did was he actually removed a bit of the cerebrospinal fluid that we talked about and replaced it with air. He removed the cerebrospinal fluid from the spinal cord, which is continuous with the brain, made a puncture typically in the lumbar area at the bottom of the spinal cord, injected this bit of air, allowed the air to diffuse up the spinal cord into the ventricular system that we talked about in the center of the head, and then took an X-ray of the head, allowing an image of general structures of the brain. X-rays, because of their sensitivity, are unable to distinguish between different colors of brain matter. They can't distinguish between gray matter in the brain or white matter in the brain. And they even have a difficult time distinguishing between fluid and non-fluid in the brain. But with the injection of this air bubble, an image was produced that allowed the radiologist to be able to see some more detail in the brain. And if we look at this picture here, we can see this is actually a woman who had been diagnosed with paranoid subtype schizophrenia. And from this image, what you can see is that the ventricles are quite enlarged. So those holes that are at the center of the head are quite large. There appears to be a reduction in volume in several of the segments of the brain that we talked about, the frontal lobes and the temporal lobes. And there also appears to be a widening of the grooves on the surface of the brain. These grooves are known as sulci, spaces between the brain. and a thinning of the swellings on the surface of the brain, which are known as gyri. So in this early 1930s image, we actually have learned quite a bit. And one could make the argument that many of the imaging techniques that have occurred subsequent to Walter Dandy's early experiments, while expanding on these findings, have not changed these findings to a great degree. The field of neuroimaging in schizophrenia was truly revolutionized by the advent of magnetic resonance imaging. Well, we're not going to go into the specifics of the physics of magnetic resonance imaging. It suffices to say that magnetic resonance imaging involves placing the head in a very powerful magnetic field. This magnetic field forces hydrogen atoms to rotate around an axis. Hydrogen atoms are found in higher degrees in tissue that includes a lot of water. And these hydrogen atoms are then displaced by what's called a radio frequency pulse in the MRI scanner. So the head is in the MRI scanner. There's this strong magnetic field, and that magnetic field is on all the time, right? It's not like they turn off the magnetic field or turn it on. It is always on in the MRI. And then a radio frequency pulse is administered that causes hydrogen atoms to absorb energy from this radio frequency pulse and then release energy. And the degree of energy that's released is a clue to where higher or lower levels of water are located in the brain. And these higher and lower levels of water allow us to distinguish gray matter, which are the cell bodies of neurons, from white matter, which are the axons of neurons, which we talked about earlier. MRI permits us to allows us very detailed, highly detailed structural pictures of the brain. We can see overlying cortical areas very well, and with a less degree of certainty, but still with quite a bit of detail, we can see subcortical structures such as the amygdala, the hippocampus, the basal ganglia, structures we've talked about already. To date, there have been literally over 100 controlled studies of the structures of brains of people with schizophrenia. When I say controlled studies, these are studies where groups of people with schizophrenia are matched to healthy controls who are typically of a similar age, similar gender, and in some cases of a similar educational level. This allows us to discern to what degree the brains of people with schizophrenia may be different from the brains of healthy controls, even when we've accounted for things like age, education, and gender. These 100 studies, or over 100 studies that have been conducted to date, have revealed a variety of very interesting findings about the brains of people with schizophrenia. Overall, it's very clear that these studies have revealed that there is reduced volume in the prefrontal cortex. Remember, that's one of the major lobes of the neocortex. And that this reduced volume is in the area of about 5% relative to healthy controls. Similar findings have been found in the temporal lobes, with about a 5% reduction in volume in the temporal lobes relative to matched healthy controls. In addition, when constituent parts of these lobes have been looked at, so in the temporal lobe, this might include the amygdala. This also might include a structure such as a major gyrus in the temporal lobe. Those differences between healthy controls and people with schizophrenia may be even larger than 5%. We also know that the lateral ventricles and the third ventricle in people with schizophrenia, on average, are 20 to 30% larger than in healthy controls. In other words, you're seeing almost a third or a 33%, close to a third increase in the size of the ventricles of people with schizophrenia on average, when you look at groups of people with schizophrenia compared to healthy controls. What's also interesting is these findings are consistent regardless of the technique we use to image the brain. So if we use CT scans, if we use structural MRI, and if we do postmortem analysis, if we analyze the brains after people with schizophrenia have passed away, in all cases, we see these enlarged ventricles and these reductions in volume in key areas of the neocortex. So these seem to be quite powerful findings. There's also been indications of increases in the size of the basal ganglia. So you can think of these 5% decreases in the frontal and temporal lobe, and you can think of these increases in structure in the basal ganglia, which again is involved in movement. These increases have been related to the amount and frequency of antipsychotic medication treatment. So we've talked about structural neuroimaging studies so far. What I want to talk about now is about functional neuroimaging studies. There are many fewer functional neuroimaging studies. In fact, current estimates suggest that there's somewhere in the area of upwards of about 40 of these studies now in people with schizophrenia. And in these studies, in functional neuroimaging, we are looking for activity in the brain rather than simply the structure of the brain. These are imaging studies that are conducted in an MRI, just the way a structural MRI study would be conducted. But in these studies, there is measurement of oxygen levels. In fact, the signal that is measured is known as what is called the BOLD signal, blood oxygen level dependent signal. And the logic behind these types of studies is that the more oxygen that's being delivered to a specific part of the brain while someone does a specific task indicates what areas of the brain are more active during that cognitive task. Functional neuroimaging studies as well have revealed a lot of very important findings about schizophrenia. Some of the cognitive areas that have been studied most closely in functional neuroimaging studies are studies of attention and working memory. Attention, of course, is the ability to focus on a specific stimulus or to be able to focus on a specific stimulus and ignore distracting stimuli, right? That's a form of attention. Working memory includes the ability to remember things over a short period of time, seconds or minutes, and even more importantly, to manipulate that information. Probably the best example would be remembering a phone number. So reciting that phone number to oneself, that would be an example of working memory over a period of seconds or minutes. But then also being able to remember the numbers of the phone number and reverse them in your head. That would be truly active working memory. So studies of attention and working memory in functional neuroimaging have converged on some very important findings. The tasks that have been used to measure attention typically been a task called the CPT, or continuous performance test, and measures of working memory have typically included what's called an NBAC task. For the continuous performance task, studies have shown that when people are actually involved in a continuous performance test where they are trying to track a target, and in this task, typically a series of stimuli are shown, many of them are distractor stimuli, and periodically a target stimuli will show up. In some cases, it's remembering, for example, to respond to a X whenever an A precedes it. So you see a series of stimuli and you respond to an X whenever an A precedes it. In other versions of the task, you're looking for a specific target, but the stimuli may be degraded visually, so it's hard to detect where the stimuli are. So you're trying to pay attention to these sort of visually degraded stimuli, responding when you see a target. In these types of studies, using continuous performance tasks, studies have shown quite decisively that one area of the frontal cortex, specifically the dorsolateral prefrontal cortex, is reduced in activation in people with schizophrenia as compared to healthy controls. This finding is particularly powerful because when this finding or this study has been replicated in first episode samples, people who've just been diagnosed with schizophrenia and have minimal exposure to antipsychotic medication, these findings are still evident. That is, when people do this task and they have schizophrenia, their activation in this area of the brain known as the dorsolateral prefrontal cortex is reduced. So again, this suggests a close relationship between a decrease here in brain activity. We had talked before about brain structure. Now we're talking about activity, but decrease in activity and performance on a specific cognitive task.